Hello, everyone. We'll be starting with today's workshop at the scheduled time in 5 more minutes. Thank you for joining. Meanwhile, we begin with today's workshop. It would be great if you could please introduce yourself in the chat box and tell us about your research background, your research interests, and what are you working on. Thank you. I would like to welcome those who have just joined the session. We'll be starting in few more minutes. We are waiting for others to join as well. Thank you for joining. And meanwhile, we begin with today's session. It would be nice if we could hear from you in the chat box about your educational background or your research work and what is your research interest. A very warm welcome to all of you present here. After concluding two successful workshops on genomic surveillance of SARS-CoV-2 variants of concern reporting and NGS single-cell data analysis and scRNA project support, we are here with another brainstorming workshop on machine learning for NGS data analysis. Today, we are going to cover practical and conceptual aspects of data exploration, statistical analysis, and machine learning in application to high throughput biomedical data using R and Python. Throughout the workshop, participants will get an understanding of supervised and unsupervised machine learning in the context of curated projects and coding challenges that illustrate methods, applications, and theory. It is designed as a combination of online resources, practical assignments, and live workshops that will be conducted online. We will also review several project examples that demonstrate successes and limitations of conventional machine learning methods and deep learning using data from public repositories. The paid participants of this workshop will be getting the access to the resources that I will be explaining in few slides. So before we begin with today's workshop, let me introduce us. We are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use analytical tools. And our mission is to make bioinformatics more accessible. Over the past several years, we have had the privilege of working with a number of organizations, developing a training program for research-oriented students, faculty, and democratizing bioinformatics, making this discipline accessible to thousands of students and faculty around the world. The research-based training programs are a result of close collaboration between Pine Biotech and numerous academic institutions that have participated in content evaluation, curriculum review, and project design. Omics Logic training has been completed by over 15,000 participants from 162 countries in over 300 workshops. And due to this fast growth, our team is working with local and regional coordinators that are helping refine the local program logistics and leverage our online training resources, adapting them to the needs of students and researchers around the world. The Omics Logic training follows a project-based learning approach, using research-grade tools to analyze data from top peer-reviewed journals. The training has been completed and is undergoing in 6 different specialization tracks, which includes oncology, infectious diseases, precision medicine, neuroscience, data science for biomedical data, and comprehensive training on omics data analysis. Honored to introduce the founders and the team members of the company. Dr. Alfred Tauber comes from a medical background with an expertise in oncology, immunology, and biochemistry of inflammation. And one of his major projects is the establishment of the Tauber Bioinformatics Research Center, which has been at the core of what we do. Dr. Leonid Brodsky is the director of Tauber Bioinformatics Research Center and is an expert in bioinformatics and biostatistics. Our platform is a result of hard work by some of the people mentioned here on the right. Julia Panoff, who is an expert bioinformatician. Avi Titavsky leads the development of the T-Bioinfo platform. Dr. Mohit Mazumdar is an experienced bioinformatician with multiple publications in peer-reviewed journals. He has and project lead for Omics Logic Training and Research Fellowship. Dr. Raghavendran Lakshminarayanan is an accomplished computational biologist with an experience in large-scale analysis of multi-omics data and data science methods in precision oncology and protein DNA structural bioinformatics. And my name is Sonalika Ray, and I'm the Omics Logic Community Manager, and I'm responsible for daily interaction with thousands of users who are a part of a community managed by Pine Biotech. So now let's gear up for today's session. First of all of you need to sign up on the Omics Logic Learn portal that will give you the access to the Omics Logic courses, which includes courses on bioinformatics, precision medicine, multi-omics, including genomics, transcriptomics, metagenomics, and epigenomics, data science that includes bioinformatics using R and Python, and example projects of different fields which you could learn from and replicate your research work. Users will be given directions to access the T-Bioinfo platform for bioinformatics processing and analysis of transcriptomics data. The platform includes demo pipelines as well as data management and analysis cloud infrastructure to run bioinformatics pipelines. Different stages of analysis are performed in different sections of this multi-omics platform. So now let's go to the learn.omicslogic portal and sign up quickly, and then we'll be starting with the workshop. Signing up on the portal will help you get the access to the resources that we're going to go through today. Let me reshare my screen. Those who have already signed up on the Omics Logic Learn portal, please let me know in the chat box by putting in a 1 or yes maybe, so that I know that you are all ready to get started with the workshop. And those who have not yet signed up, quickly go to the link that is present here in the chat box and sign up so that we can get started. And thank you, Amreen, for introducing yourself and telling us about your background. Okay, so now how do you sign up? Let me give you a quick demo to all of you present here. I'm going to re-log in for you all. Now, when you click on the login button, you can create an account for yourself just by putting in your details that are asked, or simply sign up with any of your social media accounts. I'll be doing it with one of my Google accounts. I'm awaiting responses in the chat box to begin with today's session so that I know that we all are ready with the resources in hand. Thank you, Fareen, for your response. Okay, so those who have signed up on the portal, they can click on their mail address that will redirect them to the profile tab. Now, in this profile section, you will be able to look at your profile where you can monitor your progress of various courses that you are taking, and also you will be able to download your certificates. In the profile section, you are supposed to update your profile picture. You should ensure that you have your name and title case. Having your name and title case will make sure that the name that comes in the certificate is in the right format. Please connect your social media accounts and add a brief bio about yourself. That could include about your research background or educational background if you're a student, and what are your research interests so that we know about you and can align your interests accordingly. So now let's get started with today's workshop. I'll now like to pass on the stage to Dr. Raghavendran. Those who have signed up, it's good for them. Those who have not can sign up in a while and follow along the resources with some. So over to you, Dr. Raghav.
Yeah, actually, I'll be starting, so Monica, like we discussed.
Okay, I'm so sorry, Ilya. Over to you.
Right. All right, so thank you everyone for joining today. Let me make sure I'm sharing. Yeah, can everybody see my screen?
Yes, Ilya, we can see your screen.
All right. Okay, so today I will start by giving an introduction, telling you briefly about what we do and how you can kind of benefit from what we do on a regular basis. Today is an example, and it's really designed for people that are going to be applying some of these methods to a data set. So what we will try to cover today is a little introduction to Omics Logic as a community and as a resource that you can leverage either for doing research, learning about different methods of analysis, or helping others So if you're a faculty member or you are teaching, maybe a teacher, these are resources that you can leverage. And so today's session is actually an extraction from a program that we run regularly. This program right now is ongoing for Louisiana Biomedical Research Network and for members of the National Association of Idea Principal Investigators. And this program is typically conducted as a 3 months course where we introduce statistical analysis and machine learning methods. We talk about how to design experiments and how to validate the findings. And we talk about how to apply different types of methods of machine learning like data mining and different ways to apply supervised machine learning like classification, network analysis, and others. So the program typically addresses several different important issues with machine learning. So a lot of the work to get data prepared for machine learning is an important part of knowing how to deal with different types of structured and unstructured data. It is important to understand the difference between signal and noise. And for that purpose, we have a number of exercises that are applied to different types of omics data, which I will explain in detail today. And then we talk about this challenge of data complexity or big data volume complexity, heterogeneity, and how machine learning really helps us to extract meaningful insights from such data sets and learn about the links between biology and data. So how to really get some insights from such data. So who is this for? These programs that we design are really made in such a way that anyone that has minimal background in data science can benefit from such programs. And today, I hope will be an example of how anyone with just kind of basic understanding of some of these topics can get much further ahead, can really learn and practice and get some practical experience with these complex methods. And so hopefully after today's session, you will kind of see as a perspective into the data science and machine learning, how this can apply also for other methods and other types of data. So the goal of my part of the session is to really talk about the need for data science and machine learning for biologists. I mean, why is it an important topic? What are some of the important things to consider? How to use it in your research, in your publications. If you're just getting started, how to get started. I will also talk about some of the challenges with the type of data that you are commonly going to encounter. And I will explain our approach of teaching bioinformatics to explain how you can quickly go from raw unstructured data to something that you can use. And then in the second part of this presentation, Raghav will explain how you can actually practically analyze such data using some examples. We will also talk about the logic behind the analysis and introduce the resource that helps kind of structure the logic in a way that you can follow and practice more. And then we will talk about some of the bigger challenges with machine learning specifically, why it's important to understand the methods in detail and not only learn how to run them. So why is it important to understand the meaning behind the different settings or being able to explain and validate some of the findings that you have done using machine learning? So let's start by briefly introducing data science and kind of talking about where machine learning fits into this more broad topic of data science. How does this relate to biological and biomedical data? So commonly what you will find is that data science is described as the intersection of data, statistics, and some specialization domains. So we can talk about data storage, data formats, how to manipulate data. We can talk about different analytical methods like hypothesis testing. And then typically there's a domain that you apply these tools to. And so in each one of these, you can distill them into these specific challenges. For analysis, it is difficulty of extracting signal from noisy data sets. For data, we'll talk about the differences between structured and unstructured data and the challenges of making data structured so that you can easily apply analysis methods to them. And then for specialization domain, in every domain, whether it be biological research or financial modeling, the importance is annotation and validation. So understanding some data is more intuitive than others. And in the biomedical or biological domain, we need to rely on annotation and validation using databases. Bioinformatics is typically described as the intersection of statistical analysis, informatics, and biology. And so really what it is the application of data science to the biological domain. And in this biological domain, we have several different applications like basic research, biotechnology, agriculture, genetics, biomedicine, et cetera, et cetera. So really, when we look at the two of these together, we can kind of see that the intersection really only changes by specialization. So biology is more specific for bioinformatics, and data science is just more agnostic. And so the question is, how does machine learning fit into this scheme? So machine learning is really the intersection of statistics and mathematics and computer science. And machines, in this case, are algorithms or models that we can train, and they rely on statistical methods and principles, right? In reality, many of the things that we call artificial intelligence and machine learning, they leverage a lot of the statistical analyses that have been around for a long time. What has changed are the advances in computational infrastructure, and as a result of that, some of the things that were not possible previously are now possible. And so machine learning sits at the intersection. And really what we're going to be talking today as machine learning for biological and biomedical data are the sub portions of machine learning, which are unsupervised machine learning, supervised machine learning methods, deep learning, explainable, and interpretable, right? How do we actually take these models, analyze using these models complex data sets, and then know how to explain them, know how to apply them properly, and validate that what we're finding is really true signal and not noise. So to begin speaking about this, I think we need to start from talking about data. So why is it called data science, and why does machine learning fall under data science? So data typically comes as a collection of some kind of digital signal, and data science is the study of the signal. And so when we talk about data, majority of the data that you will find is actually unstructured. And so the first 90% of your time that you're going to be spending with data is really converting this unstructured data to structured data, which is a table or a matrix or a data frame. And once you do that, you want to find associations in this data. And there are going to be many associations in the data, some of them relevant and some of them not. And so studying associations within that data are typically going to result in some kind of a model. And that model is what we will talk about today, how to differentiate between these models and how to then extract from these models some meaning. So the process from unstructured data to maybe semi-structured data, where you have many different components and you are trying to understand relationships between them, to then finally structured data. And that structured data is something that now is going to be easy for you to analyze and extract some meaningful relationships. And those are going to be what we will talk about today, because they should be reportable and actionable. Now, one of the major trends in biomedical research and biological data is the exponential growth of data that is now available for anyone to access. And so what we will do today is we will take a few examples on these data sets, but as you will find, it's very easy to find data. In fact, in just this pandemic, we just kind of did this chart at the beginning of the pandemic, and we found that the number of genomes submitted during this pandemic is many times more than in previous pandemics that have been recorded. And in particular, this is because data generation is cheaper, more accessible, and more useful. And so machine learning is especially important because now we are able to collect data very quickly And this data is large and unstructured. So when we talk about genomes, for example, these are nucleotide sequences. And so it's not easy to analyze such sequences. Why? Well, to explain why, I have this very brief example. So here's an example of unstructured data on one hand, and how we tend to look at data to be able to interpret it. So unstructured data in this case is an example for a chat, an exchange on chat. So you can see that we have a date, we have different names here of users, and we have their responses. And these are responses to questions. So for example, if I ask the question, what is your degree? What is, your highest level of education? Any person here can answer in multiple different ways. Someone will say, I have a bachelor's. Someone will say, I have a master's degree in bioinformatics. Somebody will say, high school. Somebody will say yes, right? Just because they are answering a different question. So as a result, I have this collection of unstructured data, which are just texts. And my objective to try to interpret that will be to categorize them, assign them to different groups, and measure the number of responses per group. So that makes it interpretable, right? When I look at this chart, I can quickly understand what this exchange really boils down to. And that is the challenge in structuring data that also comes from biology or biomedical research. What kinds of data do we have in biology and biomedical research that need to be structured? We have imaging, streaming data, for example, video, different types of omics data, chemical interactions, clinical records, and different types of phenotypic data, for example, cell morphology. So all of these have to be converted to a series of data frames. And the objective of preparing the data for analysis is really knowing how in each one of these are very different types of data, imaging, video data, genomic data, right? They're very different when we obtain them, how to process them in such a way that we can end up with a data frame that aligns the different samples or pieces of information and gives them features. And so this is what we're going to be working with today. Today, we're going to be looking at omics data. So when we say omics, what do we really mean by that? There are many different types of omics data, and what makes them omics data is that they are generated by high-throughput automated technologies like sequencing, and they could really be used to explore different types of biological processes. In molecular and cell biology, we typically look at how the phenotype, for example, cell morphology or function, is defined by the code of the DNA and the transcription of that code into proteins. And so we will look at sequencing the DNA, for example, nucleotides in the DNA, epigenomic regulation, how we can detect DNA methylation patterns or chromatin packing signals, and see the transcription of that code into functional proteins. So the challenge to create a structured data set is to really take that sequencing data, different types of sequencing data or different types of omics data, and make them into a structured format, which is going to be a data frame. And so what we'll do today is we'll take a few examples of such pipelines that process data from their raw format, the way that they come out of a sequencer, for example, and structuring them into a table. Once the table exists, or many tables exist, now we can perform statistical analysis and really identify important trends for biomedical or biological questions. What kinds of questions? Well, here we have a few examples, right? This is kind of translational biomedical research. Typically, you're going to be asking a question about patients. How are these patients grouped? How are they different by response to different treatments? How can I categorize or stratify diseases? Sometimes these questions are population health. Sometimes they are personalized, individual disease progression for an individual patient. Sometimes we want to take it further and we want to say, well, based on our understanding of data, how do I intervene? Are there specific targets? Are there specific interactions between drugs? Are there specific diagnostic conclusions based on these types of data, right? So these are the types of questions that we're going to be asking. Today, we're going to look at some of these examples so that we understand machine learning in the context of answering questions with data. Omics Logic as a whole, the resource, and the reason why Sonarika asked you all to sign up at the beginning of the session is because we're going to be leveraging some of the methods that are described in detail on the portal, and you will have access to analytical tools that you can try to run such analyses and get from raw data to structured to then interpretable. And the Omics Logic portal is a complete roadmap for data science and application to biomedical research data, where you will find different data sets. You will be able to explore and visualize them and understand those methods, and hopefully get enough insights and understanding about these topics so that you can then apply them to a research project of your own. And that is really the idea behind Omics Logic. And the reason why we're doing a lot of these webinars and workshops is to introduce you to the resources so that you can start learning it on your own. In the portal, you will find detailed explanations about every single type of omics data that you are typically going to be working with, including multi-omics integration. So the process of exploring these different types of regulation leads us to the need of integrating these different types of signals together. And machine learning is oftentimes used for multi-omics integration as well. And so that is why we're going to talk about several different examples like genomic data, transcriptomic data, and metagenomic data, so that we can understand the relationships between these biological phenomena and how machine learning plays a role in each one of those. In terms of applications, you can start thinking about how these are applied in oncology, infectious diseases, neuroscience, and biotechnology. And on the portal, you will find project examples coming from all of these domains where you can practice some of these methods and see how they are used by people that are experts in the field, that have published and that have produced important insights using such data. What kinds of projects are these? Some of these projects are diagnostic type projects. So for example, how do you identify patterns associated with a specific disease? Sometimes we take a look at complex single cell data and we try to extract meaning from different types of tissue composition that are typically composed of different types of cells. Or we can find associations or relationships, for example, association between microbiome composition and infection. Today, however, we will use two specific examples, and these are going to be two data sets. So Nalika, if you could already paste the links in chat to these specific projects. These projects are Modeling Cancer Precision Medicine, which is a collection of cell lines that represent different subtypes of breast cancer, and an example from TCGA, the Cancer Genome Atlas, where we will find data on liver cancer. But in addition to this, on the portal, you will be able to find many different examples, including examples from papers. Thank you. And so in the chat, you will find a link, and that link is the TCGA Liver Cancer Precision Oncology. Okay, so how do we use analytical methods to extract insights from data sets like these? The typical flow of information, for someone who is doing such research, is to have data that is cleaned and prepared, explored in the red right here in the middle, and then ready to ask specific questions or testing your hypothesis. And so as we look at these different types of project examples or data examples, we should be also thinking, why were these data generated, right? What was the reason why these data was generated? And we will find that both of these examples do not follow the standard process from experimental design, where an experimentalist came up with a hypothesis and said, to test this hypothesis, I want to now create data, right, to test this hypothesis. Both of these examples were generated without a hypothesis. They were just a collection of different types of cell lines or just patients that all fell into this great category or general category of cancer patients. And so because this is the nature of data sets, we have to rely on exploring the data sets first, or mining these data sets first, to come up with a hypothesis, come up with a question. And once we find this question, now we can turn to different types of models that will help us get inference. And inference really means that I can find some pattern and I can find the application of this pattern to the greater population. And in the context of that, a lot of people start turning to machine learning for a very specific reason. Most of these data sets do not have a control versus a condition. They have many conditions. They have many phenotypic categories. And to be able to ask questions about many categories, we have to turn, or we will benefit from turning to using machine learning, because it really provides us with that flexibility and with greater sensitivity. And because of that, the use of machine learning in biology has been growing exponentially. So here's a chart from a publication. The publication is called Recommendations for Machine Learning Validation in Biology. And basically, this paper talks about how the benefits of machine learning, the sensitivity of machine learning, and the ability to use machine learning easily has led to adoption of machine learning across the spectrum of biological research and biomedical research. And the problem that they have identified is that this exponential growth in use of machine learning also comes with a negative effect, and that is a growing number of papers that actually don't apply any evaluation of their results, because in many cases you have two major categories of users, and those are biologists that now use machine learning to identify signal, or computer scientists that really don't have the biological background. They use these tools because they understand the value of these tools. But in many cases, unfortunately, that leads to this gap of no validation and no independent test sets to kind of validate the findings. And a lot of times, no biological validation either. So as a result of that, the downside of machine learning is that it's so easy to use. It is so meaningful in many cases. You have to understand the methods really clearly, and you have to understand the settings of these methods, and then you have to understand how to validate your findings. And that is going to be a critical portion of today's and future workshops for us to not only know about machine learning, but also know how to use it properly. And with that, there are also many, many different methods, from unsupervised to supervised to semi-supervised to deep learning. And so when we look at these methods, we're going to be able to compare and contrast those methods. We'll be able to understand the benefits and the limitations of each one of those methods. And hopefully, we will also be able to understand how to extract from these methods reliable insights that we can interpret and we can use in our research. And that's not always simple, because in many cases, the biological principles of, for example, control versus condition don't always apply to the data sets that we're going to be using. And so here is an example: gene expression profiles in a particular data set that has multiple groups that cluster together don't always follow this expectation of differential gene expression, right? And these kinds of problems are going to be common across the spectrum of data sets that we're going to be using in a session like today, where we will look at different types of bioinformatics pipelines. We'll look at how to incorporate them so that we can get structured data, and then start asking questions about relationships between clinical and omics data for diagnostic and therapeutic design purposes. And furthermore, you can actually see how once these data sets are clear and these principles are clear, you can actually start scaling up the application of machine learning to a process where you automate this whole pipeline and you get interesting insights about predicting a particular feature or category using such models. So we'll talk briefly about this, how to deploy a model to production at the end of these workshops. Now, Sonalika already introduced you to the platform, and I just want to clarify how we're going to use these different tools. So I talked a little bit about Omics Logic, the portal, where you will find the data sets, you will find additional resources to help learn about these different types of data. But I also want to speak about the platform briefly. So once you sign up on the Omics Logic portal, you automatically get a profile on this portal that you will be able to run pipelines on. And these pipelines are going to be something that you will learn the logic of analysis. You'll be able to use big data, so practicing or kind of doing your research, and you'll be able to apply many different types of analysis quickly, so that you are not hindered by the technical complexities of each one of these types of data. In addition, there's a section called data mining, where you will find different methods of supervised and unsupervised analysis that we will be able to try in today's session, and then download the results as the result of the output of the pipeline, as well as scripts that run these analyses and explore them. Now, in the pipelines, you will find this logic following a color-coded explanation. So in each pop-up, you will find an explanation about the specific methods that you've used. And those explanations are also going to be consistent across many different types of methods. In addition to this, as I mentioned, once you run a pipeline, you'll be able to download the script. And those scripts could be practiced on our coding playground. So this coding playground is going to help you practice syntax and introduce you to challenges. And so once you understand the syntax, you'll be able to practice on those challenges and really understand how to write code independently, or how to at least modify code independently. We know that one of the most popular languages today is Python, and in the biomedical and biology community, R is also a very popular scripting language. So we are going to limit ourselves to just those two, Python and R. That doesn't mean that others cannot be used, but in our sessions, we're going to be using Python and R. For those of you that are new to Python and R, you can really get started with R and Python by looking at some of the coursework that we have available, just to understand the syntax, understand how these languages work. And then we kind of introduce the context of data science, and then go from statistical analysis to machine learning. And so in the coursework that kind of follows what we're going to be doing today, we have these parallel courses called BioML, and we have 3 of these courses: BioML, the logic behind machine learning, BioML in Python, and BioML in R. And all of them will explore these challenges: data wrangling, machine learning and statistics for high throughput data, unsupervised and supervised types of analysis, machine learning for data visualization, then detailed review of methods for clustering, methods for supervised machine learning or classification, feature selection and signatures, and then regression and generalized linear models, and finally deep learning. So if you want to go through all of these topics, we recommend getting started with these courses. And so briefly, I will just show you where to find these courses on the portal. So if you're on the portal together with me, and you kind of go back to the menu, you can click on the course tab and type in here BioML. And so you will find here these 3 courses that I just mentioned: BioML Logic, BioML Python, and BioML R. And if you want to participate in these courses, anyone, even with a free registration, can get started. So if you want to get started, you will find inside lessons. Many of them are free, so you can go in and get started with learning about some of these methods. You will find also that once you've started a course, you'll be able to go to your profile and see that under the courses that you have started right here. Okay, so with that, let me pass this on to Dr. Raghav, who is going to be speaking about these methods in greater detail and introduce you to some practical exercises where you can start learning how to apply these methods to the data sets that he will introduce.
Thank you very much, Elia. And and let me find ways to share my screen. Okay, I hope all of you can see my screen. Yeah, right.
Yeah, we can.
Yes, thank you. Get some. Okay. So you already had, hello and welcome to everybody for the first. You already had an introduction to how to use various omics data and then process it and then convert those unstructured data that comes straight out of the sequencer machine to structured data. So we can use and employ different types of data like genomics data, transcriptomics data, metabolomics data, and then convert them and use a set of organized processing steps in sequential order that we will often refer to as pipeline. And these pipelines are specific to each and every type of omics data that we can analyze. And at the end of the pipeline, we are going to get, or we will convert the unstructured data into semi-structured or structured data. These structured data can be employed for further downstream statistical analysis that we have. A simple example of such a pipeline setup is when you use high throughput sequencing to measure genomics data. We generally generate short reads of sequences from randomly selected or random fragments of DNA or RNA. These fragments at first have to be aligned to the reference genome. So once these fragments are aligned, and the reads can be analyzed for matches or mismatches to the reference sequences. These matches and mismatches can be and then tabulated and transferred into a set of structured data. Often, this structured data will be used in the context like comparing mutations between in oncology, comparing mutations between cancerous cells, tumor cells, and normal cells to understand how these mutations might affect important genes that are involved in tumor growth or in disease progression or in developing resistance to certain therapeutic conditions. So to analyze such data, we can prepare a table of mutability index or somatic scores for each genome position, and that is going to that will be our structured data that is coming that results from the genomics data analysis. Furthermore, you can also compile these data into a much smaller file called variance calling file or VCF file format. And this file can either be visualized by annotating the genes and visualizing the mutations, or this can further be sorry, yes, okay. Can you see my screen? Wait.
Your screen is not moving. It seems it's frozen. Yeah.
Let me wait. Let me stop and share again. Thank you. Yes, can you see it now? I think is it moving? Okay.
Yes, it is.
Okay, perfect. So yeah, so how to get or how to create this structured file for genomics analysis? You can take the raw FASTQ files and use the Taubot Bioinfo platform to set up an example pipeline, or set up a pipeline that is going to convert these FASTQ files into a table of VCF files, structured data, that we can employ for visualization or further downstream analysis. Let's go to let's cut here and go to a demo pipeline, or set up a demo pipeline in the Taubot Bioinfo platform, so that we can get a taste of how to set up a genomics pipeline or genomics data pipeline in the Taubot Bioinfo platform. Those who have credentials, please log into server tbioinfo, and when you log in, you're going to get a screen like this. And in this screen, please follow along as you have. And if you are if you have any hiccups, or if you feel any necessity to ask me to wait or proceed, we can do that and have an interactive session. So please log into server tbioinfo platform, and under mutation variant and parallel analysis of mutation variant data. So you generally have we generally have the collection of different pipelines, or setting up different pipelines and algorithms for different types of analysis, as Elia had explained it to you. So for several of these analyses, we have prepared what we call as demo pipeline, where users, you can get an experience of setting up an analysis pipeline, and understand how each and every step that is involved in setting up this pipeline is being incorporated into this analysis into the platform. And you also get access to the processed or structured data at the end of setting up this demo pipeline, which you can download and perform your analysis, downstream analysis, or practice your analysis that you have been learning in the Learnomics platform. So let's go here in the mutation variant analysis and the mutation variant data section. Please click this down arrow, and then you have several demo pipeline that has been carefully constructed or carefully designed for you. So let's check one of those. So you're going to get experience of setting up a pipeline from several different options. So in the demo pipeline, and also when you are going to set up your own pipeline, you get suggestions for your next possible steps that you can do, or that you can perform within the available data, depending upon what you have selected for the previous step. So since this is a demo pipeline, the necessary files have already been uploaded and they have been referenced. So we start it, and we start and say okay. And before selecting each and every option, you will get a short information window of what that algorithm is going to perform. For example, this algorithm is going to clean up your PCR amplification duplicates and amplification biases, and you click OK, and then you have several of these from several of these options. Now we have only one option available for our next logical step. And yes, Omnia, please. Yeah. Please share your question. You can share it in the chat box also. I think you're still muted. That's okay. So yeah, so to go forward, let's click next step in the pipeline, where we are going to, after cleaning up the PCR amplification artifacts, we are going to map these sequences or map these short reads onto the reference genome. And then we click OK. And then our next possible output is our next possible option for this pipeline or for this setup is highlighted as differential variant calling algorithm. So you click OK, and then you use somatic sniper to compare the variants that are calculated from two different biological types. Then you end the pipeline here. So once you end the pipeline, you get access to yeah, Iman, though in the same demo, you have more options. I have more options than your screen. When you click some of those options, and if you follow either of those options, you are going to get access to a different demo pipeline. One of them is going to be a visualization pipeline. The other is going to give you output like like this, a tabular output like pipeline. So depending upon what you have clicked so far, you will get different options, and you'll get you just try to click through them and understand what is what is being analyzed through the output files. Is there a link of recording in this demo? Yes, there are. Yeah, there are recordings that talks about these demo pipeline, and this Sonalika will share with you where to find those recordings. And for the purpose of today's session, let's move forward. So this is how you can perform a genomics analysis and then download the variant calling file. Since this file is too big for current session to complete download, let's move on to the PowerPoint presentation. Okay, can you see the screen? I think yes. So a similar omics data, another example for omics data is transcriptomics data. So such data, transcriptomics data, represents the mRNA expression, which is one of the very good way to analyze what is actually going on inside the cell by observing active transcription of protein coding genes. And when we normally know that DNA is first transcribed to RNA, and then RNA is translated to protein, and depending upon the conditions and the specific conditions of the cell and the stress that cell is undergoing, these expression of the protein will change and will be characteristics to the phenotypical condition of the cell or the sample that we are analyzing. So to compare between genomics and transcriptomics analysis, transcriptomics analysis is simply not a subset of genomics analysis by leaving out non-coding part. Protein coding regions generally in the DNA or the genome contain large stretches of non-coding sequences called introns, and when these introns are spliced out in RNA transcripts, then we will have different splice forms for the same gene being encoded or the same gene being developed. And depending upon the condition, depending upon the phenotype of the cell, some of these splice forms will be expressed more often than the other. And such an analysis is going to help us understand the state or help us understand the biological condition of the cell, or will be extremely useful in performing a differential gene expression analysis, which is just going to compare the expression levels of cells, for example, of tumor cells and normal cells. So how to set up such a pipeline? We can again go to the demo pipeline in the Tabot Bioinfo platform, and we will now, for this analysis, we will download the data and see what is the output data, what does the output data look like. So I will come back to the setting up of this pipeline in a bit. And when you set up a transcriptomics analysis pipeline, as a result, you will obtain a table of continuous numeric data that is annotated by gene ID and organized by samples in a given experiment. So your columns are going to be different sample names, and your rows are going to be different gene IDs given in the form of ensemble IDs or given in the form of transcript IDs. So the intersection will actually tell you the expression level for that particular gene ID and in that particular transcript, in that particular sample. So let's again go back to server Bioinfo server and set up a pipeline for transcriptomics data analysis. This time we are going to download the data to check or to get a feel for how the transcriptomics structured data looks like and what we can do from that, or what we can learn from this data. So when you go to server.t bioinfo and under the transcriptomics section, you can click this down arrow and then click either of these projects to set up a demo pipeline. Let's click the cell line project. And here also the input files, necessary FASTQ files or input files are already been inputted, and the pipeline is already run and completed. And for the educational purposes, we have developed this demo pipeline so that you can get a feeling for setting up this transcriptomics analysis. To know more about different options or different algorithms that are involved in these different options of pre-processing, mapping onto genome or mapping onto transcriptome, and quantification of these expression levels and differential expression analysis and other annotation analysis, so you can refer to the transcriptomics, extensive transcriptomics course or program, from where you can learn a lot of information. A lot of information regarding the choice of the algorithm and how to use certain algorithms for certain data sets that we have. So let's start this process again. This is going to tell you a very basic outline of how this RNA-seq analysis is going to be performed. So it involves pre-processing, mapping, and quantification of expression level at its basic level. Yes, Omnia, I think you have raised your hand. Please share your questions in the chat. I'm following the chat window all along. So after this, you perform Trimomatic, which is going to clean, or which is going to trim out the technical and adapter sequences that are necessary for identifying our samples in the pool of samples that is being sequenced together. So let's click OK and rush through a little bit, do PCR cleaning. And then we have an option now to, for example, mapping onto genome or mapping onto transcriptome. So we will now select mapping onto transcriptome option, but you can also very well select HiSat2, which is going to map onto genome process. So BowTie2 will map the reads onto the transcriptome. And after this, we get an option, or we get a highlight for selecting the expression quantification table from RSEM. So this is going to use expectation maximization algorithm to count or to calculate or quantify the expression levels of different genes, and then give it to us in a tabular format. So let's click OK, and then we end this pipeline and click OK, and you get you get option to download demo pipeline. So from here, you'll get several different options. For example, you will get expression levels of all isoforms in the cell lines, and expression levels of expression levels of all the genes in the cell line, and expression levels of exon that are specific to exons in this specific sample. And we also get, these are all absolute quantified expression levels. And we also get a link to normalized and annotated expression level. So you can click this and download these results. I have already downloaded these, and if you are following along with me, download these results, and we will open those results up in the Excel and take a peek through what we can do with these output results. So let me open up these files in Excel. So let me share everything so that it's easy for you to follow. Okay, so I have downloaded this annotated cell line and also the expression of genes in the cell txt. So let's quickly import these files into Excel to see how the structured data looks like. Okay. Can you see my screen? I think it's still visible. Let me make it a little bit bigger so that it's not very small. So as I mentioned, yes, thank you. As I mentioned, in the on the column, you will have the sample types, and on the rows, you will have the ensemble IDs, and you find the raw counts for these expression table all along here. So if we use, or if I use, if I download the annotated, normalized, and annotated table, let it open, you can quickly see the difference between these two tables. For example, this is as it has been mentioned, this is an annotated table. So what does this annotation mean? This annotation means simply the files or the samples are properly annotated. You see here, the samples are annotated by their respective names, instead of just mentioning this as the file names or the run information file names of the FASTQ files. And their values are normalized. Now we can see that their values are normalized because we don't have values like 15,000 and 16,000 anymore. Everything is between something, roughly between less than 10, and they're also negative values. So they are centered and normalized. So when will you use, for example, data in a non-normalized format and data in a normalized format? So for the downstream analysis, like statistical analysis, we can prefer to use the normalized data because most of the statistical techniques that we are going to employ will require some sort of or some form of normalization at our end. And even if we don't use the normalized data, we have to perform our own normalization on it. So this is excuse me for just 2 minutes, and I'll be back. Yes, with the back. Thank you, everybody, and sorry about that. So let's let's go forward and for example, and see what we can do with this structured data from our end. So this is the normalization is going to be at a different level depending upon what we chose. Either it's going to be count expression normalized count data, or it's going to be TPM or FPKM depending upon what we use. Yes. So let's move forward. Another type of data is actually metagenomics or microbiome data, where we are where in this specific data we are going to look at samples that contain millions of microorganisms that are represented by genomes which are very different from each other from these samples. To structure such a data that is completely varied and variable and completely varied, and a lot of variation involved with that within this data, we have to organize this information into categories and functionally relate these genes to or functionally relate the genes that are common to all these organisms, like for example ribosome, from which we can compare to a database of known sequences and then assign these genes that are common to different organisms and quantify their levels. So this is what is done in, for example, in metagenomics analysis. So since there is a lot of variability in metagenomics analysis, there is a necessary to denoise the sequencing data and map these sequencing data of found genes onto a database of known known organisms or known bacteria or known virus as counterparts, and we can classify these sequences into, for example, closely related taxonomic groups that are called operational taxonomic unit. So the sequences that are within or that form part of the specific OTUs that are called as in short, they are operational taxonomic unit, are perceived to be closely from closely related species, as they share somewhere around from 96 or 97% identity in their sequence at their sequence level. So so what do we have here? We can again set up some demo pipeline in for metagenomics analysis and download the amplicon data using data2 analysis. So for this for this, I'm not going to go through the demo pipeline, or we can actually go through the demo pipeline. We still have some more time. Let's see. Yes. Okay. Let's perform this demo pipeline, and I will show you how to get the data and what we can do with this data. So where can you access the metagenomics demo pipeline? Again, if you go to the server Bioinfo platform, down here under the metagenomics section, under amplicon analysis, then you can set up metagenomics demo amplicon analysis for the data that involves serving up, or that involves understanding the microbiome population of mouse that is fed with high-fat diet and that is fed with normal diet. So you start and use quality control. So this is going to, you can read through what each and every step actually does, and you will find that in specifically in metagenomics pipeline and analysis, a lot of denoising and a lot of normalization and batch effect correction is going on because of the nature of the data and the variability that it possesses. And these steps will be very important in our next section because we are not just going to click and set up pipelines for converting these unstructured data to structured data, where we are going to perform some of the analysis on the structured data so that we can understand how what the analysis does to our structured data when we download the output files. So here, when you download these output files, you can actually download the quality profile, or you can download the phyloseq plots. So if you download these plots and save it there, I can actually open with Chrome so that you can see this. So you will get abundance calculated by different samples, and they are properly annotated, and you also get a phylogenetic tree for different operational taxonomic unit, and that are specific to or that are color coded by different sample group of samples. So and furthermore, a lot of other downstream analysis results are also provided here. So we can get a one second. Yes. So we can get what we are going to look through, or what we are going to understand from such an analysis by performing or by going through the output file. First, what you are going to understand, what you are going to get is we will see the abundance ratio, or we will see the presence and abundance of different family or different phyla of bacteria or microbiome for different samples, and where these operational taxonomic units stand in terms of their sequence similarities with respect to each other, and how their abundance is actually how their abundance level quantifies their relation. So these information that you can get. So we again started from unstructured sequencer output data, and we are arriving here at the structured data, which is essentially a table of continuous or categorical data. So most often than not, it is continuous numerical data, which will genomics analysis signifies the mutation index or mutability index, and in terms of transcriptomics analysis, it is going to tell us expression quantities of genes under different sample, and in terms of metagenomics analysis, it is going to tell abundance of operational taxonomic unit for different samples. So that is this structured data is what we are going to use for our further analysis, which involves statistical operation taxonomy. You can obviously understand, or you can obviously get which family of bacteria, or which family of or which phyla or which order of bacteria that these OTUs represent. So that is going to give us information about the annotation for different OTUs. So this is not the only way where you can get structured data. So you can either get the unstructured data from the sequencers and then follow up with a detailed analysis pipelines and set up detailed analysis pipeline and get the structured data, or you can find structured data that are deposited into big data resources like NCBI platforms, where you will have lots and lots of structured data, expression table data that is submitted to GEO or Gene Expression Omnibus, where you will have option to download the raw sequence file, where you can create your own structured data from these raw sequence file, or you can download normalized or some sort of processed expression table, which will be helpful if you are going to perform analysis for a lot of data sets, or a large number of data sets, where we will not have enough time or enough resources to run this pipeline for a large number of data sets. So there are also specific databases and repositories like CancerX, Cancer Genome Atlas, TCGA, Cosmic, which are specific to the cancer or tumor related omics data information. And we also have Array Express, which is going to be focusing on microarray data for different data sets, and Protein Atlas, where we'll get proteome data, proteomics data, and others as well. So you'll also, if you want to focus not on human system, and if you want to focus on something like infectious diseases and pathology, pathogenesis of viruses, then you'll have specific resources for that. And this is these resources and associated repositories became extremely helpful in current pandemic situation, where they host lots and lots of data related to SARS and other viruses, and like Ebola, for which next-gen sequencing method or high-throughput sequencing method was extremely successful in tracking and developing effective combative ways for getting through this pandemic. So I will stop here and take a couple of questions if you have. Next is going to be again an interactive session, where we are going to look what we can do with the structured data in terms of statistical analysis, and what we can gain from this structured data. Please, if you have any questions, please share it in the chat. I'm following the chat along all along, and yeah. So okay, if you do not have any questions, let's go forward. Let's move forward. So yeah, we spoke about this, where we have a lot of unstructured data, and there is a huge gap between unstructured data and structured data. So we can effectively use these unstructured data to perform our analysis and develop a lot of structured data and find very impactful insights for different biological questions. Many of the biological questions and many of the high impact biological questions are actually analysis done on, or reanalysis done on data that is already been submitted to the repository. So we can also follow the steps and then create a lot of good science. So what is a structured data? We have been talking for something like 1 hour about structured data. So what is a structured data? So structured data at its simplest form is going to be a table. And there are different ways of referring to this table depending upon which programming background that you come from. And two of the most popular programming languages or background is R and Python. In both these platforms, it is referred to as a data frame. So it is referred to as data frame, and there is a underlying difference between the object data frame, which is a table of a collection of different data types, like it can be numerical data type, it can be categorical data type, or it can be binary data type, logical data type, or it can be something called factor data type, which is like restricted response to the input, like blood types and like eye color, etc., etc. So a data table, as you can see here, is a table of equal number of rows and equal number of entries in the rows and columns that is going to represent the samples and the structured data as a collection of data of different types. Of course, there are also other structured data format like matrix, which is essentially a table of numerical objects, only numerical objects. So matrix cannot contain a table or cannot contain a column or a row that is of non-numeric nature. So similarly, array is another form of structured data, where you'll have indices that are referring to elements that can either be numeric or either be character. So this in our context of moving forward, one of the important things that you want to take take into account is how to take your structured data that you have downloaded, as I have demonstrated you, from the pipeline or from any other analysis method, or from the repositories, and load it into a platform so that it becomes a data frame on which we can perform our statistical analysis. So why do we need to do this? Why do we need to perform this statistical analysis? So because any measurement, for example, since we are converting measurements of omics data into digital data, so any measurement carries information that is signal, which is of extreme interest to us, and information that is that arises due to random noise or errors, which could be masked or superimposed by the or on the signal that is of interest, depending upon their strength. So these errors are random, and of course they are unwanted, but they are always present, and they actually reduce or diminish the accuracy and precision of the measured data. And this is why we have to resort to statistical methods. And generally in statistical analysis, our aim is to find a way, or our aim is to find a function or find a model that can effectively separate this signal from the noise with appropriate, or with accounting for appropriate variation. And so in the end, when we perform this statistical analysis, we can describe the data in a for example, using a function or using a mathematical model, so that we can use this model to predict or to generalize the tendency, or generalize the structure that is observed, a pattern that is observed in the data, to future events or to general population. So this is why we have to perform some of the basic statistical analysis and resort to performing these statistical analysis methods. So let's move forward. What are the very two most useful methods that will help us in cleaning up the data or normalizing the data is normalization based on quantile normalization, and transformation of scales through log scale. So why are these important? Because this data converts, this transformation converts, for example, some of the data which are non-normal in nature, or which are not, oh, sorry, I go back, I click too much. Okay, it's not going back for me. Yes. Good. So this is not normal in nature. For example, your typical gene expression data, which can have a lot of values that are extremely low, a lot of quantity of values that are extremely low, and very few values that are extremely high. So why is this transformation important? This transformation is important because many of the methods of downstream analysis that we are going to perform rely or have some sort of underlying assumption on the data or some of the derived characteristics for the data that is Gaussian or that is normal in nature. So that is why performing a normalization or transformation of the scale to a log scale will help us even out or will help us understand the pattern in a better way. So how it is actually helping us, for example, for a non-log transformed comparison, our comparison between any two genes can look like this, where many of the information is actually populated very close to zero, and very few of the information is actually falls along the line, which can be extremely misleading, because when you convert the scale into log scale, we actually see a very good acceptance between, or very good correlation between the expression level of these two genes. And by performing quantile normalization, what we actually essentially do is we take the expression levels of different samples, and then we match their match their medians or match their average expression levels, so that all of the samples follow the same or similar trajectory or similar distribution. So by performing a quantile normalization, we are actually forcing our data to be normally distributed, so that our analysis can hold real meaning and value. So in the spectrum of a gene expression table, for example, as often we are going to relate to gene expression table, because in the following slides, so we can actually separate this spectrum into three parts, where at its lowest end we have signal and noise intermingled with each other, and it's going to be extremely difficult to separate or identify which is signal and which is noise. So we are not interested in some of the data that is present here, and in the and in the center, where we will have normal gene expression levels. And this is going to be very relevant for us, because we have to set up expression levels or expression quantities at the normal scale, with which we can compare the expression levels at the highest end. So this is the significant signal which we are extremely interested in, and which we will be using for comparing the expression levels of samples of different nature. So for this to happen, we should not normalize this significant level. So that is why when we are going to perform a quantile normalization, we have to mention, or we have to use a threshold beyond which or above which the normalization is not going to take any, or is not going to have any effort. So we will set up this pipeline in a short while, and I will tell you why such an analysis or why such methods are important. For example, these methods are important to test our hypothesis, or to test our interpretation, or to test a hypothesis that what we are about the pattern that we are finding in our data. For example, usually for such a statistical analysis, we will have condition A versus condition B, and we will be extremely interested in identifying what are the things that are, depending upon the structured data, what are the patterns that are found in condition A that are not found in condition B. So by this analysis, we can actually tell whether the samples in condition A and samples in condition B are significantly different with respect to each other. So this is going to happen using, for example, sorry. Okay, this can actually follow a lot of exploratory data analysis, which is going to help us learn a lot about our data. So our next live session, that is day after tomorrow, is going to have a hands-on session or hands-on tutorial about how to perform these exploratory data analysis for the data, where we will explore, or where we will use some of these techniques like using scatter plot or using box plots or bar plots to generally understand the underlying pattern in our data, and that can be used or leveraged to develop a hypothesis or to generate a hypothesis that we have to test, or to generally set up a contrast or comparison level that we can use for our use, for example, a t-test analysis to understand the variation between two groups, two sample conditions in general. So such an analysis is going to give us, for example, patterns that are specific to one sample set with respect to the other sample set, and from this we can infer, or we can make a lot of biological meaningful biological implication and inference why such sample, or why this difference may be biologically relevant and useful, and what are the important, or what are the significant genes in terms of transcriptomics data analysis and their expression levels that are specific to one set of samples, and not specific to the other set of samples, and how their expression pattern vary, whether the expression patterns of these genes can be employed in serving up or in using this as a biomarker for these specific samples. So so let's, for example, take this further, and I will pause here to ask if you have any queries or not. If you do not have any queries, then we will move on to perform some unsupervised learning and machine learning methods and understand much more about our data in general. So the necessity to do statistical analysis is I think is very clear for you, and what we are going to learn from this statistical analysis is also clear. So until now, we have been discussing about comparing samples, like comparing two sets of samples. So if you have, for example, let's say 3 different conditions, then we can obviously perform 2 separate comparisons for these 3 different conditions. But what happens when you have multiple conditions, and we cannot just keep on performing all pairs of comparisons? So for those times, or for those specific analysis methods, then we have to rely on to higher or more advanced statistical methods like machine learning methods, where you can employ unsupervised learning or supervised learning to generally understand, or to generally identify patterns related to multiple different sample types, or multiple different identities that are found in the sample. So I will pause here to ask if you have any queries or not. If you do not have any queries, let's go forward. The next session is going to be again an interactive session, where I'm going to follow you along with the courses that are found in Omics Logic, and also set up a couple of pipeline so we can understand how in general we can use unsupervised learning methods to understand and to identify patterns in our data. Is everything clear so far? So if everything is pretty okay, then let's move forward. So generally, we as I mentioned, machine learning can be separated into 2 main categories of methods, like unsupervised machine learning that focuses on detecting patterns in data, and the second one is supervised machine learning, where once we have a labeled data, we can use these labeled data to train a model, or mathematical model or algorithm, and then use this trained algorithm to predict or classify a new data set. So so the main idea of applying these machine learning techniques is to understand the relationships between the features, or between the genes in our data, and between the various assumptions that follow, or that define the data, and to model or mathematically model this, and to learn from this, so that we can better characterize the samples accurately in terms of unsupervised machine learning algorithms, or to predict any new sample to be belonging to either of the category that the learning algorithm has chance to learn about. So in the unsupervised machine learning algorithm, so what we actually have is we generally start with the data set, typically a table of elements like your structured data. So for the representation purposes, let's understand, or let's believe, or let's assume that this table of elements are represented as these shapes. Then we can use clustering method, for example, different types of clustering method like hierarchical clustering, k-means clustering, or PCA method to identify groups based on the similarity of these shapes or based on the structure that you find in your structured data. So such a method actually results in separating the data and assigning samples to groups of elements that are present. So this separation, or this segregation of patterns usually use or employs dimensionality reduction method, which is a transformation of your variance that is found in your original data to newer dimensions, or to transform linear or non-linearly transform dimensions, which we will learn a little bit more about in subsequent slides. So it is possible to understand, or it is possible to generally, okay, go back, sorry, yeah, it is possible to my PowerPoint is actually taking long time to respond. So the result of such clustering or unsupervised data analysis machine learning algorithm will be to give us patterns in the data, so that we can group our samples or group our data into specific groups and correlate these groups with the phenotypical characteristics that we generally associate with. So one of the most used unsupervised machine learning algorithm is principal component analysis. So what is PCA? What is principal component analysis and how it is used? So in PCA, we use a simple approach of using components or principal components or transform components that is going to explain the variance of our data in the best possible way or in the best possible dimension. So for example, here is the data. Let's say here's the data that is with extremely low variance, or with the that we can infer because the samples, or because the data points are clustered together, are very close together. So for example, this is a data with a higher variance than compared to this data. So but in spite of this data having higher variance, the variance that is present in this data is not explained very well by the axes of the choice that we have. The x and y axis does not explain the variance that we generally see in this specific type of data. So what you can actually do is you can actually draw a vector through the multidimensional space that maximizes the variance of the data, and this vector is called principal component. So this is not going to be a straightforward line that is going to be represented in a 2D scatter plot, but you can visualize this in such a way. So in a multi-dimensional space, if you draw a tensor, or if you draw a vector, or not a tensor, if you draw a vector that is going to maximize the variance that you see in or observe in your samples, so that will become, or that will form your first principal component. So you can follow this up with maximizing the variance in the second dimension. That is going to form your second principal component. Go there and play. Take this to play. Sorry about that. Yeah. So for example, we will visualize this by changing or converting our scale, or converting our axes to represent the principal component and not the original axis that we chose to represent this from. So such a transformation is going to yield separation of our samples based on their variability, or based on the maximum variability that the samples are described in our example data set. So let's quickly cut short here and then demonstrate a way where you can set up a principal component analysis pipeline or PCA pipeline in the Taobot Bioinfo platform. So for this, if you have a subscription to the platform, then you can follow along with me. If not, then please go through the where is this? Okay. Yeah. So if not, please go through the demo as I show you, and then we can use, or we can follow along after you have gained the subscription. So for example, in the BioML course that Ilya was talking about, then we have a dimensionality reduction methods or dimensionality reduction method lesson. So the lesson is structured in such a way that first we will learn about the basics, or first we will learn about the background of what is a dimensionality reduction method and how that is going to help us visualize the data, or how it's going to help us overcome what is generally referred to as a curse of dimensionality, which is nothing but, let's open up the data. The access to the data can also be had, or can also be taken from these links. Let me share this link to you in the chat window. If you have access to the learn omics logic, then sign in to the omics logic, and then if you click this lesson, and you can access this lesson. If not, then it's going to go to the home page. So don't think there is something wrong with the data or something wrong with the link. Then you can do this. Okay. So if you log in, then this lesson you can access this lesson. So you can take these three data sets. So these three data sets are actually aptly labeled as input for PCA with the ID, or input for PCA without labels and with labels, etc., etc. So they actually tell you, or give an explanation about how they are describing. Oh, excuse me. Just a second. Sorry about that. Just minding my kid. So so let's start with one of these data set and then perform a PCA analysis. Let's see. For this specific lesson, I think they start with the input file that is not labeled, or that is not. Let's see what do they mean by not labeled. So let me share everything so that you can see what I see. So I have downloaded these three data files, and these three data files, we can visualize these files as this ones. Okay. Let's see if we can visualize it here. It's easy to visualize it here. Yes, but let's use Excel. One second. Yeah. So first, let's start with the file that is mentioned as expression tables without label. So if you open up this file, we'll wait for the file to load up. Yes. So it is going to be your normal data where you have this file. We had our introduction to this specific file where we had the samples as columns, and you have your ensemble IDs as rows. So if you go to Taobot Bioinfo platform, so in the platform, you can set up a principal component analysis using different ways. We can either use unsupervised analysis section, or you can use utilities section, or either of these. So when you use unsupervised analysis section, you use a file upload, and then you upload this file, a specific file. We upload the file without labels, the one that we just had a look, and it will, yeah, after uploading, let's go down and we have to start setting up a principal component analysis visualization or pipeline. So there are different ways of two different algorithms that will perform the same principal component analysis, and PCA, this option is going to give you results without visualization, and this option is going to give you result with a visualization to PDF format. So let's continue, and if you start, if you are with me, then follow along with me. Let me give you a link for the input file. Where is the lesson? So the link for the input file is in this lesson, where you can download this file and so without the IDs, it's going to be here. Let me copy this link address and I will share it with you in chat. Yeah, you can download this and then set up a pipeline along with me. So so our pipeline is here. Sorry, here. Yeah. So once it is uploaded, then you start it, and where is the OK button? I think it's masked by the Zoom. Okay, it's not there. Let me reset it and do it one more time. So essentially, yeah, if it is taking too long, I think many of us are accessing the Tubboat Bioinfo platform at the same time. So what I wanted to tell is, if you use the data without label or without any annotation, the data that has only sample information on the first row, so we are going to set up a PCA pipeline just like this, where we can start with uploading the data from the unsupervised analysis section, and then use PCA-R library, and then finish it. It's a very simple pipeline, and you are going to employ, or you're going to be asked for the option of number of principal components to be calculated, and if you want to scale your data, or if you don't want to scale your data, or if you want to center your data or not, and if you want to transpose your data. So transpose your data would generally mean, so in our data, you see, we have okay, where is our data again? So in our data, we generally have samples along the columns and gene expression or ensemble IDs along genes along the rows. But we are interested in understanding the patterns between the samples, not between the genes. So we have to transform this file to perform a PCA analysis in R. So that is what it is asking. So that option we have to mention. So yes, we want to transform this, and then save up the parameters, and then download, use this to run the pipeline. So this is going to give you a PDF output of the PCA, principal component analysis, that we have performed. So this is the PDF output. Okay, let me ask you, what do you infer from this file, from this output? So let's take a bit down, zoom out. So what do you infer from this output? Anybody? Can you tell?
PC1 explains 15.13% of the total variability, and PC2
Yes.
12%.
Okay, perfect. So the top, so the first principal component is going to explain you 15% of the variance that you observe in your input data. And the second principal component explains you something around 12%. So when you add up PC1 and 2, it's going to cover something around 27%, which is not excellent, but we can take it depending upon the looking at the segregation of the data that we have. But can anybody explain, or can anybody stretch even further to interpret this file? What do we have here? Do you see any pattern, and whether the pattern is actually reflected in the colors or not? So generally not, because the colors are everywhere, and if you see, the colors are the dots are actually colored by each and every different sample. So that is not a good grouping method, or that is not a way in which we are going to which is going to be extremely informative. Yes, of course, you can see by relating to the darker, colors that are like blue and pink, which are obviously in this area. So these samples are going to be in one cluster, or very close to one cluster, and these samples are going to be in another cluster that is far away from this. But generally, this is not good because the data is not annotated. So by annotation, I mean the data is not reflecting any phenotypical characteristics that is associated with the samples. So that is what we will see in the file, in the file that has the labels, that has the groups associated with them. So the file that has the groups are, there is only one difference between the file that has the or the table that has a group and the table that does not have the group. That difference is, how can I make this bigger? Okay. So that difference essentially is there is another row right below the samples, right below the sample row, which tells actually which groups these samples by their phenotypical type. For example, this is non-malignant, or these are the control samples, and these are the basal type samples, and these are the luminal cancer types, and this is Claudine low, and etc. as such. The power of such analysis, or the power of such annotation, you can actually check, or you can actually visualize when you run this platform with this specific output. You will see. Let me share my presentation again. It's here. Okay. Yeah, you will actually see at the end of the analysis, you will you can have, okay, let's go to the next slide and come back to this slide. Yes, you will see the samples, they will be colored by the groups or by the annotation that we have chosen for, or that we have opted for. That becomes extremely important because then we can relate to the sample segregation on the PCA to the with the phenotypical characteristics of the sample. So can anybody interpret this principal component analysis output for me? So what do you see here? And I hope you can read the key labels that are down here below here. So can you please tell me? Can anybody tell me? So I will explain this or expand the acronyms. So LIHC would are the liver carcinoma and liver cancer, and BRCA is a breast cancer, and there is a control, and there is adjacent normal and tumor samples along them. Yes, Dr. Iman tells there's a clear discrimination between the groups. Yes, there is a clear discrimination between the groups, obviously, because the colors occupy different quadrant that we can see, and I can, yeah, maybe some more can be extracted from this analysis. What is the what do you see if you relate to samples, relate the samples with their annotation? So the first striking feature that I see is principal component 1 explains the maximum separation or maximum variation as observed, explains the maximum separation between the samples, and the samples are separated by their tumor types. So see this, green and blue are liver cancer cells, and red and orange are breast cancer tumors, breast tumors. So yes, so they are separated by the tumor types. First, that's a prominent separation, and that's a maximum variation that we observe in our input data set. And the principal component 2 is very useful in separating the subsequent tumor cells or tumor types into adjacent normal and tumor samples. So these are the important implications or phenotypical associations that you can relate to the principal component analysis if you use annotation by grouping method. So let's go back one slide. So Sylvia, yes, good association, yes. So what can you learn from this principal component analysis output? So apart from the obvious things like principal component 1 tells it is approximately or almost 22% associated, and principal component 2 tells almost 8%. And what can you also find from this analysis or from this output file? Anybody? I'm expecting some more answers. Please go ahead. Please go ahead. It does not matter whatever whatever the output is.
Blue dots are more correlated, while orange dots show much more variance. That is why they are more spreaded.
That is terrific. That is excellent observation. So the blue dots are the ER samples that are annotated as ER samples have very or a very low degree of variability among them, and the triple negative samples have very high degree of variability among them as characterized by the spread along the principal component 1. And there is one glaring observation that anybody can make. What is that? Yes, who is that? Iman, Dr. Iman. Yes, there is one very far outlier. So we have one outlier, very far outlier here, and we have generally a decent separation between the sample types along the principal component 1, and very obvious outlier. So principal component analysis is not only an unsupervised machine learning technique or unsupervised analysis. It is also employed. It is also employed as a part of exploratory analysis for this specific reason. One, to identify patterns and underlying segregation of samples and patterns of samples, and two, to identify the presence of outliers in the data. So if we see such outliers, which for most of the data you will obviously see, and for the other plots you are not seeing such outliers because those data sets are curated for the purpose of understanding the logic behind the algorithm. So just a moment. So that those are the ways in which principal component analysis can be used. So let's move ahead and see what we can learn from this one. So principal component analysis is just one such method to reduce the dimensions of your data, of your structured data. So why do I call reduce the dimensions of the structured data? Because remember, in the expression table, the one thing that I never talked about is the size of those structured data, size of the expression table. And why I have not spoken about that, because those we will discuss in detail about the size and about how to check different statistical summary of the data in on Thursday. But when you generally see an expression table, and that is going to give something around 21,000 or 20,000, we can in theory populate 24,000 genes with their expression patterns. But you are anywhere good to see in terms of tens of thousands of rows. So So you have essentially tens and thousands of dimension to describe the samples and their variance and their differences and their patterns. So you cannot obviously visualize, or you cannot make a plot that is going to take, or that is going to take into account of 1, 2, 3, and 4, 5, or the tens and thousands of different dimensions. So when you perform a principal component analysis, or an analysis like that, so what principal component analysis does is by generating components that is going to describe your data in the order of most variance explained, you are essentially reducing the number of dimensions that is required to explain maximal variance that you observe in your data set. So very soon your data is going to have your data is going to cover most of the expression, or most of the variance explained by principal component analysis through something like 10, 12, or even 15. The principal component analysis is sufficient to explain 90% or over 90% of the variance that you observe in the data. So that is why they are regarded as these linear and non-linear transformation of dimensions are regarded as dimensionality reduction techniques. And principal component analysis is one of the extremely useful dimensionality reduction techniques, because it uses the explained variance to show or to accurately represent our data. But sometimes, or most of the times, this way of using the explained variance to transform our dimensions into principal components might not always work well for our kind of data. For example, you remember we spoke about metagenomic sequence data being extremely variable and has high degree of variance, and another characteristics of metagenomic sequencing data is the presence of lot of null values. So these are the different challenges that you will encounter in your statistical analysis steps. So when you have a data, so when you are going to analyze a data that is not populated with a lot of null values, so you can generally use principal component analysis to describe or to transform or to reduce the dimensions. But the moment that you have a lot of null values or a lot of values, zero values, then unfortunately you cannot use, our principal component analysis is not going to be accurate enough for your for the data transformation. So in such conditions, you can use multidimensional scaling, or NMDS, or principal coordinate analysis, or T-SNE methods, or T-distributed stochastic neighbor embedding. So these methods are going to actually are going to understand the samples based on their variability and based on their relatedness. So or based on the neighborhoods that they can establish in the data. So these methods can actually help us visualize the patterns in, for example, in extremely variable data, and data with lot of null values in, like metagenomics data, single cell RNA data. So sorry about that. Yeah, I will be there. So in that instance, we are going to use something like T-SNE and NMDS to display or to describe how connected or how closely related different samples in our data set are. So most usefulness of these non-linear transformation dimensional reduction methods like NMDS, principal coordinate analysis, and T-SNE are for metagenomics data and for single cell RNA seq data. So so those are for statistical analysis regarding principal components using principal component analysis. So if you have any queries so far, please let me know. if you do not have any queries, let me stop sharing and start sharing my screen with the about Bioinfo platform and see what we can learn from the principal component analysis that we can use, that we can employ in the environment or in the programming environment R. Okay. Okay, TSNE and NMDS again, please. For example, let's put it this way: the main goal of multidimensional scaling or NMDS is to is to study the information and collapse this information from multiple dimensions into just a few. So they can be, for example, they can be visualized or they can be compared. So principal component analysis uses variance that is studied in the dimensions to segregate or to separate the patterns in the samples. But methods like NMDS and TSNE actually use the distances or primarily the Euclidean distances between the samples to cluster them in the nonlinear dimensional space. So that is why you will see the distance-based methods like NMDS and TSNE are much more flexible to accommodate outliers than you saw with the principal component analysis, where that one big outlier was just sitting there like a sore thumb. So yeah, but in the event of, for example, TSNE and the NMDS method, you can see that NMDS is very closely, it resembles close to principal component analysis, but they are coordinates analysis, and that uses, for example, rank orders, and these rank orders are they depend upon Euclidean distances. For example, they calculate the distances for all pairs of samples and use these distances to rank these samples with respect to each other, and then they perform clustering on those samples. So that is how principal coordinate analysis or NMDS is going to give us ordination or give us sample segregation. But while TSNE is actually, it uses something like nearest neighbor, or it uses connectivity based method to establish patterns in the data. And this connectivity based method actually depends on some of the initial steps. So whenever we are going to discuss TSNE, we will be discussing perplexity. So perplexity is like, how to put it? I mean, yeah, if you want to have, I think perplexity is closely related to whether you want to have microscopic or macroscopic pattern, or whether you want to observe microscopic or macroscopic pattern, or global or local pattern. Let's put it this way: global or local pattern in your data. So I think higher perplexity is going to give you a lot of local patterns, and the lower perplexity is going to give you a lot of global, global pattern. I'm not very sure about that, but one is going to give you global, the other is going to give you local. If anybody has more information on which one is which, they can correct me. But what it actually does is it starts by assuming all the samples are connected with all the samples, and then it calculates the neighborhoodness or nearest neighborhoodness connectivity for these samples, and then it keeps on kicking out neighbors based on their based on the number of connections each and every neighbor has, and thus we have, or we get not a very uniform pattern or cluster of samples, but this TSNE is actually capable of producing or capable of identifying clusters even in very complex form of data, as you can see here. That is why TSNE is being used to study patterns within different cell types in single cell RNA analysis and other analysis stuff patterns. So that is a little bit more about TSNE, but today is not about TSNE and NMDS, so that's why I have not prepared a lot of other slides for TSNE and NMDS also. Let's see if we can address it in the Thursday session or not. So let's quickly stop sharing and go to Taobot Bioinfo platform, where I want to demonstrate you how we can employ PCA analysis, or where you will find information on PCA analysis to be performed using R as a programming language or R as a programming environment. Okay, there is a new chat. Okay, so in for example, we saw how PCA can be set up using Tauber bioinfo platforms unsupervised machine learning algorithm option, where you saw parameters like these important parameters. Now, if you are going to use R as a tool, or you can also use Python as a tool, we can obviously we have a lesson that is going to first take you through what is principal component analysis and the necessity to do such an analysis for the data that we have, which we have covered in Excel here. So it is the code based lessons are structured in such a way that you are first given an introduction about what you are going to learn and the background for the analysis to be performed, and then you are going to learn the code in a step-by-step format, where you will have a couple of videos demonstrating the power of such analysis and methods, and you will have a section where the code is broken down into line by line, where we are explaining you the syntax behind the code and the necessary options that are associated with each and every syntax. The goal for today's session was to go through each and every line, but I think it's taking a long time. So I urge each and every one of you to go through this this code. You can this code lesson. Let me copy it and give it to you. It's in everyone in the meeting. Yeah. So if you are signed into Tauber, into Learnomics, then you can access this lesson. So you go through the necessary libraries and packages to be loaded, and then these are the syntax that you have to deal with, and how you are going to, for example, read, or how you are going to read the data table, the structured data that is sitting in your computer as a text file, and how you are going to load that table into a programming environment. That is where the first step we start with. That is where the challenge lies. And once we have the table loaded and placed into an object that is usually a data frame, then we can perform a lot of calculations on that, and related calculation that we want to perform is principal component analysis. And principal component analysis requires the data to be in a matrix format and not in data frame format. If you wait, let me share everything. I think I don't have yeah, let me share everything so that you can see my screen and see what I can see. So if you remember our structured data table, usually they are without groups, or they are without labels, and what we have is a typical structured data with the first column as gene IDs or ensemble IDs. So this can be loaded into R's environment or Python's environment as a data frame, but the principal component analysis requires not a data frame but a matrix. So to convert this table or this data frame into a matrix, we have to first leave, or why this one is coming again? Yeah, we have to first take this out, or use only numerical values that are in our data frame, and leave out the ensemble IDs or the gene IDs that are associated with every row. But don't just delete it, we save it into a different format. So, and then we can convert our data table into matrix format, which is absolutely necessary for performing principal component analysis. As we have spoken in detail about performing principal component analysis, for understanding the variability across the gene or across the gene expression pattern, we have to transpose our data transpose our data, so that for performing principal component analysis, the genes are along the columns and the samples are along the rows. So we do this transformation, and then we simply use the PR-comp function to calculate the principal component analysis. So the principal component analysis that is calculated can be used to visualize using multiple different options. So all of these all of these options that you can use to visualize and to color code and to understand the patterns in the data, you will not have such a flexibility if you are going to employ automated analysis like Taubot Bioinfo platform. But why did we insist on doing this analysis on the platform, where you will get an idea of how your data is going to get transformed from a multi-dimensional or a very high dimensional structured data into a dimensionality reduced principal components file, where you will have we can use a PC1 and PC2 to perform a scatter plot and understand the patterns in this. So so obviously when you plot scatter plot, it's going to have it's going to come up like this. Then we can use the colors to represent which samples are from which groups, and that annotation is possible. And I urge all of you to go through this code one by one. You can have you can understand how the principal component can be extremely effective in understanding the patterns in your data, not only about the samples but also about the genes that best explains the different different segregation of samples. That can also be understood in this loading plot. And we come down, and as it is the case with any programming environment, we don't have just only one option to perform any defined analysis method. So we perform the principal component analysis using PR-comp library or PR-comp function, but we can use the ggplot or gg45 library to visualize the output in a much richer environment and with lot of options, so that you can so that your visualization is easy. And this is what gg45 and autoplot is what is being used by the PCR library that we have set up for our principal component analysis. One thing that I forgot actually to tell you is, when you run PCR library, or when you run PCR library option, and when you finish along with the output, what you are going to get is a zip file, and that zip file is going to here it is, PCR pack.zip, and this zip file is going to contain the R code that was used to develop, or that was used to generate this output. So if you understand the logic behind performing PCA by going through this lesson, you can essentially go through that R code and then change only few of the components to visualize or to associate our data set with either the subtypes of the tumor, or with associated risk factors, which you can see in the TCGA projects that you were given link to earlier. So we can employ this auto plot, or with gg45 or this PCR library option to use the same calculation for PCA, and then visualize that with both with the subtypes of the tumor and also with the risk factors associated with the data. So this code block is actually followed by an extremely useful code or section called practice the learn output. So what you actually will find here is, you'll find small blocks of consoles which you can use to run, which we can use to practice your codes. So at first, usually the first code is actually given in completion. So all the necessary codes are complete, where you just run and get the output. So you can open up the output and check check the different visualization. But what actually follows here is, if you just type run, it is not going to run. It is going to give you, okay, this also runs, sorry. Yes, this is learn the code, and this is practice the output. And in this practice output, you are given these code blocks where you can run for different settings and understand the output. What I was interested in telling you is the assignment section where we have actually left some of the obvious samples, some of the obvious syntaxes incomplete, like this one and like this one, so that you can compare and contrast the syntaxes and the codes that you have learned so far from practicing the code and from learning the code, and put it up here and complete this sentences so that you can run and get the satisfaction or get the understanding of how each and every code is going to work. So now when I run this as such, it's going to give me a lot of errors, and I'm not going to get the job complete output. So these are all very simple syntax completion exercises, which you can easily finish up in less than 15 or 20 minutes if you follow along with this code. So this is what I wanted to show you, how intuitive and how useful it is to go through the R codes in the analysis in the R code environment in Learnomics Logics platform. So that is what you're going to get, and you will also get an option to go beyond the basics, where you will find multiple not so straightforward and complicated ways to generate much more useful plots, which you can explore them and then practice them also. And in the end, when you have a Python lesson, not R lesson, you are given a Colab notebook, which you can use to change or modify the codes as it suits your data or as it suits your analysis methods. So this is the going through the sections of learning the code and practicing the code is very, very useful, because along the way you will set exactly the same parameters that you used to set in the pipeline analysis. So that is going to be the theme for all the data science or all the machine learning based analysis, where you will have the logic explained in the and the power of this analysis explained in the Taubot Bioinfo platform setting. And then once you have understood what this analysis can do to your data and how it can be helpful, and when you are interested in learning how to achieve such methods and how to analyze these methods, then you can use the associated R lesson or Python lessons to understand the necessary codes that are required to generate such output methods. So I will stop here and ask if you have any queries so far, or I will quickly move on to clustering. Yeah, very quickly. So if you have no questions to follow, then no questions to ask, then we will go through with with what we have so far. So so far we have done unsupervised machine learning algorithm analysis using principal component analysis, and spoke a little bit about a lot about dimensionality reduction techniques. And clustering analysis is actually at its simplest form is a task of grouping a set of objects in a way that the objects of same group or objects that are found in one group are more similar to each other than those are in the other groups. So the important aspect of this is this approach seems to be, for example, deterministic, as it absolutely tries to find out the association of each and every group that the sample is associated with. I mean, what do I mean by that? If you have an obvious outlier, it tries its best to accommodate that outlier into either one of the clusters, even if it means, or even if it reduces the accuracy of the clustering. So that is why this method is deterministic, and we have to be extremely careful in applying this method to some of the data that is filled with outliers. For example, here, we know that from this grouping, these two are different clusters from the color. I think if you can look through the purple and the orange color, so there are purple color dots here and orange color dots here and one green color dot here. But the clustering analysis can very easily cluster all of these together. So, and and very easily cluster these together, where we will not have any problem, because most of the participating samples are dots are from the same color or same group. So that's what I meant by clustering analysis being deterministic. So this is what, this is what it finds. Obviously, we will have some inaccuracies along the intersection of two clusters, along the section where we have where the boundaries are not defined properly along the two clusters. So what does the data and how does the clustering is performed from the structured data that we have so far? So we have already been going through this data for a long time. So let's take a look at our data. So we know that we can associate our data with the different groups, and groups in this case are clinical subtypes of breast cancer. So where each gene has a profile. So each gene has a profile for along the samples that along across the different sample types. And these data set, as it can be seen, as it can be yeah, I'm sorry about that. So I muted and went to attend my son. Sorry about that. So these genes, as we see, will have different patterns across different samples. So we cannot obviously plot a bar plot for each and every sample. So in order to understand the patterns, we can actually use or employ a clustering method so that the gene expression patterns of all the samples inside those inside the specific clusters are defined as a group that belongs or that expresses the gene expression in specific patterns. So that is what we are going to find. Like, for example, when we plot these bar plots along, this is a limited data set, like only 15 genes are given here. So when we can find patterns, obviously, but we can not do this for thousands and thousands of genes that we have. So there are different clustering methods and algorithms that are available for depending upon the data that we have in our hands. So I'm not going to go over all of these methods. I'm just telling you, giving you a heads up that we have different methods of clustering, and each and these methods will actually depend upon the data that is going to be used as subjected to clustering. More often than not, it's a very it's an acceptable practice to compare 2 or 3 different clustering methods for different parameters that one can use to judge these clustering options. So hierarchical clustering is one of the popular approaches to perform clustering, because it actually has a lot of options that allows clustering at different levels. Usually this algorithm does not require an expected or, for example, expected number of clusters, as it is often with the k-means clustering. So it starts actually with establishing hierarchy based on closeness, or based on the samples that are closely related or not closely related. Let's say if we have our samples organized into some space or an imaginary 2D plot, let's say these samples are positioned somewhere in some perspective that best represents the data variability. So the algorithm will first find let's say 2 closest objects and then link them together, and then this is called a cluster in hierarchical clustering algorithm. And then those 2 linked objects will become one cluster, and that will be represented by only one object or one sample. And then it does this iteratively to include all the remaining data points into either of the clusters that it had already or that it had calculated so far for the data that we have. So how does that one work? So the hierarchical clustering effectively does not require the user to mention number of clusters beforehand, like it is usually the case with k-means clustering, but it gives the output in the form that is called a dendrogram. So this is a dendrogram where the relation between samples are represented as a representation that is not very dissimilar from phylogenetic tree dendrograms. So here we can use this this dendrogram to identify how closely related two clusters are, and this is how the clustering is actually performed in hierarchical clustering, so that until all the samples are used up into belonging to one or the other cluster. So this actually gives us a hint on what are the necessary. Let's not go over this and each and every step of this clustering method. I will quickly skip through. Yes. So what we actually have is, it calculates, to do this, it actually calculates the pairwise distances between the clusters, and then it constructs this using these pairwise distances, a distance matrix from where the algorithm can identify pairs of clusters that are closely closely situated or located, and then it continues to merge all of the samples into clusters until we get one cluster here. So after the cluster analysis, or after creating these matrices, or after getting the dendrogram, one has to mention, or one has to specify how many clusters do we expect to see from our data analysis. No, it is not related to NMDS, because NMDS it also calculates, yes, in some sense it also calculates Euclidean distances between the samples, but in some sense you can say that it is little bit related to NMDS, but NMDS is structurally different from hierarchical clustering in a way that hierarchical clustering averages out the distances between two data points that NMDS doesn't do. It just ranks each and every sample by its relatedness or by its distance to other samples. So, but hierarchical clustering, after clubbing two or after making or creating a cluster, it calculates the centroid for that cluster, so which is the average of all the samples that are put together. So NMDS doesn't do that. So how to set up clustering in Taobot Bioinfo platform? It is under unsupervised analysis in data mining area, and you only need the structured input file to go through setting up setting up the hierarchical clustering. So again, for hierarchical clustering, some of the important set parameters that you have to set is to define the linkage method, and to define the distance with which it is calculated, and to tell how many clusters do we expect. But this number is actually used only after creating or after calculating the clusters or after calculating the dendrogram. So this is not employed beforehand, before creating the clusters like in k-means clustering, but this is done only after creating the clusters, which can be changed after calculating the clusters. So when we move forward to linkage method and distances and different parameters. So what is actually linkage? So a linkage is actually finds how two or more clusters are actually linked together, and how they are for example, how their distances are calculated. When you have an average linkage, it is going to calculate the distances between these two clusters by taking all of the cluster members into account. But when you have single linkage, that single linkage can be distances between two closely related or two closely located members, and complete linkage would mean distances between two far apart situated members, and centroid linkage is another method where it is actually calculating the centroid. So depending upon our sample, we can use different linkage method to either to get effective segregation, and to calculate different clusters, to calculate the clusters for our samples. So it is often useful to use different distance method, like Euclidean distances, that is the most used, but Manhattan distances is also used, and other distance methods to employ how the clusters arrangement changes with respect to different linkage methods, and sometimes each one of them will have their own place in the data analysis. So this is, selection is based on best, best cluster visually, and how do we evaluate cluster? That is the next step, right? We got cluster that looks like this, the dendrogram that looks like this, and how do we evaluate the accuracy of this dendrogram? So obviously we have to, this is as I mentioned, if you use for different clusters, after creating dendrogram, the cluster hierarchical clustering identifies how many different types of clusters that is present in our data or in our data set. So how to employ clustering, or how to, in another effective way, how to interpret the clustering? So essentially we can compare actually how different, and compare and contrast how clustering is related to dimensionality reduction techniques like PCA, and how it can provide us with the very useful insights about the data. So in this PCA analysis, you can obviously see that luminal groups are, just excuse me for a moment. I'm sorry about that. So to interpret this PCA plot, we can easily visualize and interpret that the luminal samples are the one that are far apart, or that are segregated completely from the other types of samples. So that is the essential separation that we see in the dendrogram or hierarchical clustering also. But in addition to this, we can also see this separation can be represented in the two major clusters that are observed in our data set. So in addition, we can also see several other for example, separation when you use when you employ most distant samples, like here the non-malignant sample is the one that is the most distant from the luminal sample, that you can see here also. The non-malignant sample is the one that is most distant from the luminant sample, as seen from the PCA variance. So this we can see, and so one of the good way, as I mentioned, to test what we see here is one of the good way to test clustering is to, to compare the phenotypes that is associated with the sample. But can you tell me from this data, we have not been interacting for a long time, can you tell me from this plot, what is one thing that clustering will give you, or clustering output will give you, that PCA will not? So far in the past 2 or 3 slides, I'm actually comparing and contrasting the outputs of PCA and clustering. And what is one thing that clustering is actually providing you with that PCA will not give you? Selection is based on, but no more queries. I think everybody is tired. So let's skip and move forward. So clustering can also give you distances, can actually give you estimated distances between the clusters based on different parameters that we have calculated so far, but PCA will not give you the absolute distances. So that we can use. And so clustering can actually tell you some of the surprises that you can see. For example, in this specific case, there seems to be an outlier, but this outlier is actually a luminal sample that has been completely misinterpreted or misidentified with, and that is referred to in this publication. So clustering can also clustering can also help us in identifying samples that are oddly segregated or located, and then understand their validation based on their segregation due to the gene expression pattern. So clustering is one of the widely used data visualization technique, where we can see organized patterns in heat maps, and we can see gene expression data that is organized as a upregulated or downregulated set of genes, and these can be employed to identify clustering in our samples and visualize clustering in our samples. So far, we have been talking about only one mode of hierarchical clustering. There is also other types of clustering. So this is what I said. So different gene expression patterns symbolizes or effectively creates different clusters that are used for visualization and other unsupervised machine learning algorithm analysis, where you can identify patterns within the data. So this is also a data mining method where multiple different clustering methods are available, and depending upon the method, you will see different clusters being produced. And I hope you can see, yeah. So what I wanted to represent here is, it does not matter, or in some sense, the choice of the clustering might be obvious if you have a very good data, but most of the clustering will actually have glaring difficulties in identifying or in understanding the boundaries between the clusters, and locating the outliers that is present in the clusters. For example, the deterministic manner in which the clusters are generated requires this particular data to be part of this cluster, but we can definitely see that this is completely far away and this is an outlier. So that is not the case. So these are some of the difficulties in using clustering analysis for our machine learning or unsupervised machine learning based techniques analysis. So these 2 points we have to keep in mind. We should not have outliers in our sample, and that can easily be avoided by performing an exploratory data analysis like PCA. And another thing, we might have some difficulties in the intersection between the clusters. So when we have clusters that are closely related to each other, so you are bound to find some inaccuracies there. So one of the way to combat that is to use different linkage methods and use different distance calculation methods to find commonality between these different techniques to understand or to characterize these clusters. And there are also more sophisticated way to, for example, quantify the correctness or quantify the accuracy of these clusters, which is not being discussed in this section so far. So until now, we have been talking about clustering based on gene expression patterns that will cluster samples into groups of into associated groups. But you can flip it around and you can transpose it and find and cluster the genes, so that why it is not going. Yeah, quick. Yes, and cluster the genes to find co-expressed clustering, or genes that are generally co-expressed together, or genes that share similar expression levels. This is also widely used for example, in single cell RNA analysis or in metagenomics analysis to identify common gene expression patterns and other cases. So yeah, this is also widely used case. So for example, when you use clustering across samples, it can straightforwardly yield a very meaningful output, where you see different groupings based on the gene expression pattern of samples. But depending upon your data, the clustering for genes might not yield a very straightforward or meaningful separation. So you can use k-means clustering 6 or 7 or 8. All of them will divide this pattern into different clusters, but we obviously do not see or we do not see different clusters in this specific distribution. So what do we do in that case? So we have to employ different cluster methods. For this, k-means is not a best clustering option as visualized here, and you have to employ something like spatial clustering or other UMAP and other clustering methods to understand the clustering options that we have. So I wanted to go over how we can use clustering, or how we can go through the lessons of clustering in R and also in Throughput Bioinfo platform. But I think I briefly touched upon how we can go through clustering methods in Throughput Bioinfo platform. Since you get the hang of how our lessons are structured, I think you can also go through the clustering lessons in R, where you can find the associated lessons. And I can take some questions if you have any. This has been a long session so far. So let's quickly take a couple of questions. And we finished today's data. What type of data used for input of clustering? Okay, that's an excellent. We usually use some sort of normalized data, not raw counts. We use either log transformed or quantile, either log transformed or quantile normalized data. Okay, and in some sense, you don't need to normalize the data, but normalizing the data, you only need it for further other analysis, because most of the clustering method is going to calculate the Euclidean distances, which is completely, which is not in a way it is dependent on the scale that you have. So it is better to use log transformed data. It is better to use log transformed data. Yes. So that is generally accepted as a good method, because we normally, or we usually visualize the clustering on PCA, and for PCA you definitely need to scale and center the data in some form or the other. So a log transform data is the most used for clustering methods. So any other queries? Yeah, so go through the asynchronous lesson tomorrow, and day after tomorrow we are going to deal with the further exploratory analysis, and I think we are also going to deal with supervised machine learning techniques. If there are no questions, then I will leave the podium to Sonalika to take over. Thank you very much for your participation for a long time, and I look forward to meet you all in day after tomorrow on Thursday session. Sonalika, if you
Yes, sir. Thank you so much for the wonderful session. And I would now like to reshare my screen, and let's discuss about the other sessions. And if you have not registered yourself, then let's find a way how you have to do so to get the access to the resources for a month, and also be a part of second and third day of the workshop. So I'll just take a minute to reshare my screen, and I just want to make sure one thing, that this workshop is definitely for 3 days. We had a wonderful first day today, and I hope that you all had a great start and were able to learn more about machine learning and its use for biomedical data analysis. So now to just a second. Raghav sir, can you confirm if my screen is visible? Okay, thank you, Vipra. So I was telling you about the workshop page and how you can register yourself for the workshop and get the access to Onyx Logic Learn resources that includes the coursework, the BioML coursework, R and Python that Dr. Raghav went through today, and also Ilya discussed about it in the beginning of today's session. And also get the access to the Bioinfo server and work with your data set and on using the machine learning utilities and get a good hands-on practice. So now to get an access to the other 2 days of the workshop and to the resources for a month, it's important that you register yourself. Today was the first day, it was introductory day, and that's why we thought let's invite everyone who's interested to learn about machine learning and have a look at what sort of workshop this is all about. So this is the program page, the link of which I've shared in the chat. And this program page will help you look at the content, or let's say the session details that will be covered in the workshop. And then on the on the program page itself, you will be able to look at the recordings of these sessions that we'll be having after being processed. I'll be uploading it up on the program page, and you'll have it with you for a month that you can go through. And obviously it's not easy for all of you to absorb so many things in just 3 days, so just go through these resources like recordings, coursework, the server practice pipelines at your own pace. So you'll find all these resources, the recording, and also the associated resources that you have to go through up here on the page itself. Now, to enroll yourself for the workshop, you have 3 levels: beginner, intermediate, and advanced. In the beginner level, you will get the access to all these resources that I just discussed, the sessions that we just had, and be able to share questions with Dr. Raghav. And if you would like to have a one-on-one session, maybe, then I would suggest you go for an intermediate type of subscription that will help you get in touch with Dr. Raghav one-on-one and have your questions answered. And also we have the symposium coming up. It's on March 31st and April 1st. So taking the intermediate subscription will help you get your seat saved for symposium as well, and you will be able to be a part of the symposium and listen to the great talks that we're going to have from various international institutions and universities and industries as well. And so, talking of which reminds me to just quickly go through the symposium page so that you don't miss out on this opportunity and you can become a part of the same. So this is the symposium page. The link is in the chat box. And on this page, you can see what we are going to focus on, right? We have mentioned the categories of our symposium very clearly here on the page itself, and the speakers that we're going to have, their institutions, what they're going to talk all about. We also have great researchers and scientists coming from LSU, Amity, and the industry experts from Google, Faba, Prima's Life Sciences, and we'll be having some great talks, right? And if you would like to, let's say, be a part of symposium only, then I'll also share the details with you so that you can register yourself for the symposium and save your seat. And also we have poster presentation competition in the symposium, and the last day to submit your abstract for the same is 20th March. So if you are interested to present your research work in the symposium, you can definitely submit your abstract, and I'll share the link with you in the mail definitely, and you can proceed your application in the form that will be given in the mail. The third option is an advanced level. Now, taking an advanced subscription will help you get an extra time to work on your research proposal, go through these resources, discuss with Dr. Raghav, and come up with a good research proposal that you can work on as an independent research project, and definitely have a good outcome out of it, either a preprint or a publication or a review article, or at least a report that you can submit in your college if you come from graduation or a post-graduation background, and or you are in a final year, you will definitely require to submit a project report maybe. So having this subscription will help you in the same. So these were the 3 levels, and if you are interested to pick any one of these and become a part of the workshop for a month, then you can reach out to us. I'll put my mail address in the chat box. It's marketing@atomicslogic.com. And the sales team or the business development team, including Sparsh, she'll be available when you mail on this email ID, and she'll be able to help you out with your enrollment and take your application ahead. So that was all about the workshop and the symposium. If you have any questions, please put it in the chat box so that I can answer. Any questions? Okay, so if we have no more questions, thank you so much, everyone, for being a part of the session. And we'll be looking forward to meet you tomorrow. Thank you so much, Dr. Raghav, for such a wonderful session. Looking forward to your session tomorrow. Thank you. Bye-bye.